Hello and welcome to the September 2016 Get Started with CyVerse webinar. This 1-hour webinar is a way for us at CyVerse to introduce our user community to the various tools, services, products, and platform that we provide. So I want to welcome you. My name is Jason Williams and I'm coming to you from Cold Spring Harbor Laboratory in New York. Over the next hour, I'll have a chance to not only show you some live demos of the products and materials that CyVerse provides, but also answer your questions. Before we get started with the presentation, I want to make you familiar with some of the technologies that we're using in this Adobe Connect room to come to you for the webinar. First of all, importantly at the bottom is actually a place where you can answer or ask questions. So please, at any time, whatever your questions are, or if you have any type of technical difficulty, you should see that space at the bottom where you can go ahead and put your questions. Off the screen to the side here, I can see any questions that folks might have, so please go ahead and do that any time. Also, on the side of the screen here, you should see that there are some important links and downloads. So you can get a copy of the presentation. You can see some important links to the CyVerse websites and tools. At the top of the screen, there's also a little button with 4 arrows in it, and you can click that to change the size of the screen. So you can actually make the video portion full screen. And that's going to be important when I do some demos on my computer. It will be a little bit easier to see my computer screen that way if you make it full screen. Also, when you move your mouse over the video itself, you can change the quality of the video. So if your connection is not that great, you can go ahead and actually alter the connection speed, maybe reduce it from HD to something that is a little bit better on the bandwidth. So with that said, the format for the webinar is I have a quick presentation of a few minutes just to give you an idea or a background of what CyVerse is as a project. Then I have a series of demos, both on how to share and manage your data with CyVerse, how to get started using the discovery environment to do analyses, and also working on the cloud. So if you have any questions or issues during that presentation, please just let me know. I also see a question from Margaret. So if you have an issue with the video, I would just recommend that you adjust the bandwidth in case anything is breaking up for you. If anybody else is having any kind of issue or audio or anything like that, please let us know. Usually if more than one person says something, then we might know that the problem is our end. But at this moment, it seems like we're broadcasting at good quality. But let us know if that doesn't resolve or if anybody else has a problem. Okay, so to get started here, I just have a couple of quick slides that I want to show you, give you a little bit of an idea of the history of the background of the project. Okay. So the first slide here is about the evolution of the project itself. We want our users to know this. About every month we have somewhere around 400 or 500, 600 users signing up for the service, often by word of mouth, but they may not always know exactly why or what the history of the project is and what our mission is and what we're actually doing. So through this project, well, we actually have a vision which we list as transforming science through data-driven discovery. The mission of the project and the way that we actually help you to transform your science by making it more data-driven is by designing and developing and deploying cyber infrastructure. So that's essentially what we build as a service to the community. And we also train scientists in how to use that cyber infrastructure to address their individual scientific problems, questions, research projects. We have more than 30,000 users. We have a large collection of data. There's hundreds of publications, courses, and discoveries that have all been enabled by what CyVerse has delivered. But one thing that's important to us in terms of our history is that we've not always been called CyVerse. In fact, it's just this year that we've adopted that name. The project started in 2008 as the iPlant Collaborative, which some of you may have heard of before. And with the success of that project after the first 5 years, we were renewed and we took on this job of not just empowering plant science, but really all life science. And now today in 2016, we really could say that our mission is to empower data-driven discovery throughout life sciences. So no matter what type of research question you're coming to us with us, it's something that we have applications to support. The other thing that's important about our project is that we're funded entirely by the National Science Foundation. So, pardon me, we are not a company. We don't sell a product per se, although sometimes I'll use that word product. We're actually your colleagues and your collaborators. So it's a $100 million investment that we've provided, and it's freely available to the community. The purpose of the project is to really spur collaboration. So whether your collaboration is national or international, you can make use of CyVerse freely. In fact, for our international users, everything is completely available, no matter where you are accessing the system. Also, we do ask and remind our users to please cite us as they do make discoveries and publish, because that is a way that we demonstrate the impact of what we're able to provide thanks to NSF funds. That large investment, again, means that you are not paying directly for this product. Okay, I've used the term cyber infrastructure. I just want to quickly make sure that everybody's on the same page with me in terms of what that means. When you come to CyVerse, you have the ability to access data storage, you have software, also high-performance computing, and finally, people. So all of those things, we would say, are the ingredients behind cyber infrastructure. And they're not something that necessarily you haven't encountered before in many cases, but it's really organizing those different systems into something that works well and that allows you to solve problems of size and scope that otherwise just wouldn't be possible. So really our big win at the end of the day is that when a researcher comes up to us and is telling us that, hey, you know what, I could not have solved this project, or I could never have thought about tackling this type of research question unless I had the CyVerse resources. That's a win for us, enabling you at whatever scale you are to do more than you could have ordinarily if you didn't have access. So platforms, tools, data sets, and also today is an example of teaching and training. Those are all the things that make up CyVerse. And when we put that all together, we really would say that CyVerse is built for data. So whether you're coming to us with plant data, animal data, data about climate, ecology, and biomedical data, any of those types of data sets, really CyVerse is built for data. It's largely agnostic to the type of research questions. You'll not only find the tools for managing and using, working with those data sets, but you'll also find the software and the tools to do the analysis. So it's really all of those pieces of the puzzle that we've sort of put in one place for you. Okay, I want you to just take a look at this next slide. Gives you a broad overview of what we call the product stack. Not everybody will use every single thing at every single moment, but it's good for you to know that you might have come to CyVerse looking to do one thing, but there are multiple things, and maybe you'll grow to use some of those. So on the top layer, we have what we call our products, which are Atmosphere, the Discovery Environment, DNA Subway, BISC. I'll mention some of these, although we won't do demos. We won't have time to do demos on all of those. Beneath the surface are various services, things like authorization that keep your data secure and safe, but that allow you to log in to each of these services with a single set of credentials. The data store, which allows you to move data back and forth between these services and, of course, between you, the user, and the CyVerse environment. Searching for those capabilities, obviously if data is not discoverable, then it may not be useful. And then at the very bottom layer, we have the foundational capabilities. And these are even accessible for users who want to work through our APIs, the high performance computing, the storage, the cloud systems, really depending on what level you're at. And most of us are bench biologists, I would say. And so we're operating up here at the sort of point and click, sometimes also using the command line. But also if you're a developer and working in that area of bioinformatics or systems, you can actually make use of even the sort of hardware resources fully customized. I'm almost done with this presentation in terms of the background, but let me just clue you into how CyVerse was built. And remember, this is coming from a project that started back in 2008. Really, we knew from the beginning that it was going to be a community project. We had to gather a large amount of community input. We still do, but certainly that shaped us in the beginning. And so when you come to CyVerse, really still today, even though there are many, many data sets, a lot of data types, a lot of things are catching up, like image analysis, but really genomics has, and for the time being will be, a very, very large and significant driver of data within the life sciences. And so when you come to CyVerse, you'll find tools for assemblies, for RNA-seq, for variant calling, looking at variation, ChIP-seq, looking at epigenetics. All of those different things are really really well-established pipelines within CyVerse, and all determined by the community. This is important for me to note because essentially, as a tip of the way that CyVerse functions, what you see is there because the community asked for it. And if you don't see something, let us know, because often that type of input will shape what comes next or what's available. Okay. The last little bit here about sort of the history and the organization of CyVerse is that we are an entirely collaborative and virtual organization. So CyVerse per se, the original product or project, is a collaboration which represents the efforts of folks at the University of Arizona, where there is a principal body of folks there. Also at the University of Texas Austin, Texas Advanced Computing Center, so TAC. Here at Cold Spring Harbor Laboratory, where we provide drivers for science priorities as well as education and outreach and training, and also at UNC Wilmington. An additional sort of spin-off project, which is funded by BBSRC, is CyVerse UK, which is a collaboration of the various universities and also the Earlham Institute, in which case they wanted a local copy of those resources, and so they were able to leverage what we built and build it out to their needs. Okay, so moving now on to the more practical things. I thank you for indulging me. Maybe you know a little bit more about CyVerse and how it works. To draw on an analogy, we think of ourselves to some extent as Lego blocks. We've built many of the pieces that you will need in order to make your workflow produce the results that you needed to produce. And really, if we've built 85 or even 95% of you want, then you also have the resources to put those last few percent in, customize it, make it into what you'd like it to be. So what are those pieces? Well, now to go into a little bit more detail, number one, I think, is the data store, which is the ability to store and move and manage any size data set in practical terms. Every user has an initial 100 gigabyte allocation. But if you need more data, that's certainly available to you. You can have larger allocations. We just need a little bit of justification from you. The data are automatically backed up, and we've made it really easy to upload, download, and share. And I'll be showing you some of the features of the data store, get you started with that in just a few moments during our demo. The discovery environment will also be demoed today, and this is a web interface. It's a platform where you can run almost any type of bioinformatics application. It's integrated with both the data store and also high performance computing, and it's also user extensible, so you have the ability and opportunity to add your own applications. Atmosphere is cloud computing for the life sciences, so it gives you simple one-click access to hundreds of virtual machine images. You can customize your software setup, and it's also integrated with our computing and data resources. The science APIs, this will not be demoed, but it's something that's important because there are bioinformatics developers who certainly would like to get their hands on some of the resources and customize it and build what they need. So it's a science as a service platform. You can define your own compute, storage, and resources. Large elements of what we have for those people who are developers are what we call Dockerized. So it makes it really easy to bring your applications via Docker into the CyVerse systems. You can work really with the scientific codes and workflows that suit you. DNA Subway also we won't demo today, but that's educational workflow for looking at genomes, barcoding, and RNA-seq. There's a whole rich set of resources for working with bioinformatics in the classroom. And importantly, a large number of the experiments that you analyze are also combined with wet lab techniques. So it's really great for teaching and bringing students into the world of bioinformatics by letting them analyze data that they've generated. It's also great because what we call inquiry-based experiments, course-based research, cures, all of those things are really very powerfully supported by DNA Subway. Bisq is our platform for image data. You can manage those image data. You can look at and analyze data in hundreds of different image formats. So that's really wonderful if you're working with that type of data. And those are the major platforms that I wanted to discuss in terms of just giving you an overview. You may not use all of them at once, but it's good to know what's available and what's there and what to look more into. Unfortunately, we only have time to cover demos for just the first 3. And so the first thing we're going to start with is a bit of a demo on getting your data into CyVerse. Obviously, if you can get your data in, that's sometimes the first big step. Let me also remind you that if you have questions, I can see those off screen, so please feel free in the bottom there to send your comments. Also, this might be a good time if you're going to watch me do some of these things on my computer to make the video full screen, which you can do. You can enlarge it at the top there. Okay, so getting data into CyVerse. As I mentioned, we have the CyVerse data store. You can store any type of file that's related to your research. So you may have image data, sequence data, whatever. You can move that data, those files, seamlessly between different platforms. You can automate those transfers. You can share with your lab members, collaborators, and also different communities. There are 3 main ways, I would say, that you interact with the data store. The first 2 are point and click, so using CyberDuck and the discovery environment, so I'm going to talk about those, and then also on the command line. The discovery environment is great for everyday data management, so simple upload and download for small files. You may have some larger files that you can use the discovery environment to upload, but we really recommend CyberDuck for large files that you may have. You may also need to import data from URLs, and we can do that as well on your behalf through the discovery environment. This covers most of the basic needs, but there are some size and speed limitations. CyberDuck is really almost, I would use the analogy of like Dropbox, where you can drag and drop files and folders. There's no size limit. You can also do some file editing and previewing, and it's really easy for the desktop functionality. It's more like a desktop file system. There are some unique features of the data store, such as the ability to manage metadata and permissions on your files, and you cannot access that directly through CyberDuck. iCommands really lets you do everything. So if you're a command line user, and it's really not even if you haven't used the command line, it doesn't take that much to get iCommands working. It allows full flexibility, so you can script and automate transfers, you can access from a terminal. It's completely customizable. The only disadvantage is if you don't know the command line, you may need to take a little tutorial to get yourself started and try it, but it's really not too difficult. Okay, so I'm going to give you a quick demo first on CyberDuck and iCommands, and we'll come back to the discovery environment in a bit because we're also going to analyze data there. Okay, so on my, well, let me take you first. Here's my desktop there. Let me take you first to the CyVerse homepage, because this is often the starting point for anything that you'd like to do with CyVerse. So please take a look, see what we're up to, look at the news. But first, let's go down here to the Learning Center. And from the Learning Center, we're going to go ahead in this second link here where it says Managing Your Data. Okay, so you have all of the information that I'm going to cover. You don't really need to remember what I'm saying. I'll also say that this is all recorded, so this will be published to YouTube shortly. But we're going to do CyberDuck here, so I'm just going to focus on that. And I'm going to click on Using CyberDuck with a CyVerse account. That will take you to a set of instructions on the CyVerse wiki. Let me make that not so big. And essentially, you can follow these instructions to download CyberDuck and get it installed. There's 2 things you need to do. You need to actually go and download CyberDuck, which is a standalone program that we don't actually manage or create. And then you also need to download one more file, which is sort of like a bookmark that makes it specific to the iPlant, or really CyVerse data store. Now, once you have CyVerse installed, and I already have it on my computer, this is what it looks like when it's connected to your data store. Sorry, I can't make it look too much bigger. Hopefully on your full-size screen you can see it. So this is the CyberDuck window. If I start a new, let's see if I can do that here, a new CyberDuck browser, when you first start CyberDuck, if you've set it up according to our instructions, it will just have this bookmark there saying data.iplantcollaborative.org. You'll double click on that and give it a few seconds to connect. Now obviously I'm already connected in this other window, so I won't wait for that. It usually takes a few seconds to connect. But once you are connected, this is my CyVerse data store. I have folders, I have files. If I want to move something to my desktop, for example, I can just drag it over. A new window will open, and it will go through the process of moving that. I'm on wireless, so things might be slightly slower, but essentially it'll tell you a download. And in fact, I have my upload and download history of what's going on. I can see the progress of any existing transfer. And if I want to move a file, let me just go ahead and create a file on my desktop. Okay, this is an empty file that I've just created on my desktop, and if I want to take that file and move it to my CyVerse data store, I just drag it over, and that will also appear in my list of transfers, and we'll go ahead and move. I can certainly drag it into a folder. I could move files and folders that way. This also, just to give you a little bit of statistics that's important to know, we would expect on a decent connection that you could move an order of 100 gigabytes in about 30 minutes. So that's relatively acceptable, I think, for most people. So you should be able to move a decent amount of files from your local computer or from a hard drive straight onto CyVerse with not much more effort than dragging and dropping, as I've shown here. Also hidden, not directly shown, once those data are copied, they're also automatically backed up. When I get to the Discovery Environment demo, I'm also going to show you how you can manage and get some additional details. But just as a matter of getting the files on and off the system, using CyberDuck is a great way. Just don't do it on wireless, as I'm doing right now. Okay, so let me return back to the computer and show you how to do this with the command line, and then we'll move on to looking at the Discovery Environment, because here it really is as simple as drag and drop. Okay, so if I want to move things, I'm going to close that for a bit. If I want to move things using the command line, I will go, let me show you where that is. Go back to managing data, and then it talks about this other program called iCommands. And so there it says with a CyVerse account, so I'm going to go ahead and open that. So iCommands is a series of, or is a set of tools that you can use within CyVerse. You're going to have to download iCommands for your particular operating system. There is a version for Windows, but in my experience, I don't know if it really has been updated yet, so that has had some issues to me. But for everyone else, you can use iCommands. Apparently with the update, if you're really familiar with Linux, you can also use the different installers that are available to you here. Once you have iCommands brought down, there's going to be a few questions that you need to fill out, or a few configuration items, like the name of the host, the port, the zone, all of those things are right here. So you're going to have to fill that out the very first time. I'm going to go ahead and open a terminal, and let me open one that's a little bit easier to see with contrast here. All right, and if I want to start iCommands, it's the initiate command, and that just asks me to enter my password. iRods is the name of the underlying system we use for our data store. Okay, I've entered my password. Okay, and nothing happened, which means it's good, it accepted that. And now if you're familiar with things like Linux, an ls command would list the contents of your directory. And so to use the I command, we do ILS, and that will list the contents of my I would say iPlant previously, but now I would just say my CyVerse data store. It would list the contents there. Okay, let me just cancel that in the background. Okay, but just as you saw a moment ago in CyberDuck, although this is in the command line, those same files and folders that are here on the left-hand side in CyberDuck are what I'm viewing now in my terminal. If I wanted to move some folders or files, well, let's move a folder from my terminal onto my desktop. It would be an iget command, and I want to be recursive. I want to see the progress and be verbose and the time. And let's move that VCF, and I'll move it onto my desktop. Okay. And so that command, and you don't have to guess at the commands, I'll go back and just show you quickly in a moment, is all listed in documentation. But then what happens is it will start making that, moving the files and give you a progress report of what's going on. And as you can see on my desktop, that folder is already there and being populated with the files that I'm downloading. In fact, if I go back to the documentation For I commands, moving down a little bit, there is the next link to learn about using those I commands, and it will have all of the details on how to do everything from change a directory, alter your permissions, metadata, etc. All of those commands are there for you to use. Again, that requires just a little bit of familiarity with the Linux style computing or Unix style computing, but it doesn't take too much to learn. It's quite useful. Okay, so what I've just showed you in about 5 minutes or so, I think, is that it's really easy to move data up and down, move data into and out of CyVerse using any of those tools. What I'm going to do now is I'm going to move us into the discovery environment. We're going to keep talking about data because I need to talk about, well, how do you actually share those data once you have them uploaded? And then we'll move on to how to analyze it. Okay, so let me just go back to my slides, which will orient us for a moment, and then we'll continue with that. Okay, so the discovery environment. As I mentioned, it's a platform. You can run almost any bioinformatics application that you would normally run at the command line. It's integrated with the data, which we're going to focus on right now, and high-performance computing, and you can also add your own applications. The first thing we're going to look at is the data window, where we upload, download files, share via URLs, or share with other users. We'll talk about applications, which allow you to run your applications, build workflows, install new applications, and we'll talk about analyses, where you actually can do some science. So let me open that up first, and I'll come back to 1 or 2 slides and show you about analysis later. Okay, let's get this out of the way. All right, so I'm going to go back to the CyVerse homepage. And from here, this time I'm going to go ahead and click on Discovery Environment. And at the bottom here, it'll say Log into the Discovery Environment. So I'm going to go ahead and say yes to that. It'll ask me for my CyVerse username and password. And I'll make this slightly bigger. Okay, and this is the discovery environment. It opens up the last windows I used. I'm going to close that just temporarily. As I mentioned, there are these different panels for looking at your data and managing and sharing, using applications and doing analyses. On the right-hand side, there are notifications of things that have completed. There's some preferences. There is a link to our user forum where you can ask questions. But I'm going to go ahead and focus on data. And it's the theme or the metaphor here is really like a desktop. So I click on a folder, and everyone has a home folder, which is the same as their username. And that contains everything that we just saw a few moments ago in the CyberDuck and also at the command line, where you see a list of your files or folders. When you click on individual, let me click on a file, for example. When you click on a file, not only do you see some information here about when it was modified, the size, but on the right-hand side there may be some additional information about that particular file. If I want to share a file with somebody, there's some ways that I could do it. One of the really quick ways to just share an individual file is to go up to Share and say create a public link. Okay, and I just got a little error there. Sorry, excuse me. I was waiting for something. So what will happen is this window will come up, and it gives me the ability to create a link. And I'll just wait for a second, and it creates this link, which is a URL. Now I can copy that link and hit Command-C to copy it. And now if I gave that link to anybody, let's go up to here and just click in that link, it would start a download that they could download that individual file. And they do not have to be a user of CyVerse, so it doesn't require that whoever you send this to is has a CyVerse account. It's almost, again, I think in Dropbox there's the functionality where you can share a link with somebody and they can just go ahead and download it. Okay? When you create a link that way, you have to recognize that that URL is basically public. It's discoverable on something like Google. When you give it, you send it in an email to somebody, you post it online, someone else could share that link. So if you have files that you consider sensitive, you probably don't want to share them this way. Or when you do share them, you want to come back to this same dialog and click that little minus sign I can make it slightly bigger, but you click that little minus sign to delete the data link. Once you delete that link, by clicking on it there, it will no longer be usable, okay? So that anyone who would go back and click on that link again, they would not have access to the file. Of course, if they already downloaded it, then they have access that way, but once you delete it there, that same link wouldn't work, okay? Now, that's good for individual file sharing, but often we have large data sets that we want to share. There are ways, if you have a large data set, let's say that's part of a publication and it just needs to be available and you want a DOI for it, and we can actually do that for you. Those things may end up in our data commons, which is part of what you see here in community data, and those are accessible outside of CyVerse. But for now, I'm going to focus on what do you want to share a large folder or collection of data internally. I'm going to go back to my home folder. This way of sharing that I'm about to show you actually does require that who you're sharing with has a CyVerse account, but as you saw, a CyVerse account is free, so it's not a gigantic deal for someone else to have that CyVerse account. So let's say I want to share a particular folder. I go up to share, and this time I say share with collaborators, and then it's going to load this window. It'll show me already if anyone has access with this folder. I think I can already see that no one else has access. So I can search for users, or I can put users in. So let me just type in the name of somebody I know. As you type in the name of the person that you want to share with, or you put their username, you should find their name if they have their account, and also their affiliation. And then you can decide how you want to share it with them. So we allow you to assign, this is a very similar thing to the Linux permissions, either read, write, or own permissions to that data or data set. If I go back to the documentation that we were in earlier about sharing data, just go back a couple steps. I may actually need to go back to the wiki, CyVerse homepage, Learning Center, and managing your data. Okay, so back here, oops, actually it's not managing your data. I think there's actually one specifically for sharing those data. So in the discovery environment here, it says, it's going to take a look at that. I just want to show you, or I could even, there it is, sharing files and folders. Just what those permissions mean. So if you're curious, you know that there's documentation on that, okay? Here it is, permission levels. So if you give read access to somebody, they can read those files and they can even download them, but that's it. If they have write access, they can change the name of the file. Info type is just what we call it. If it's a FASTA file, then it's a FASTA info type. If it's a BAM file, it's a BAM type. And then own is the highest level of permission, and you have to be careful because they could even move those data from where you put them. They could delete the data, so you wouldn't want to give that permission unless you're absolutely sure. Okay, so going back to discovery environment, if I wanted to give Cornell here write access, I would click done. And he would get a notification in a few seconds that that file has been shared with him. Since really it's changing the permissions, this is instant. There's no waiting for hours for something to be moved around. Whoever you're sharing it with will have virtually instant access. If I didn't want to share this with Cornell anymore, I'd come back here, I'd see that it's shared with him, and I could click that X, and then click done, and then he would also get a notification that it's no longer being shared with him. Okay? Things that are shared with another user appear in the shared with me folder that's here in the discovery environment. If you click on that, you'll also notice that there are lots of files or folders that are already there, because there's some things that are shared with everybody regardless. It's sort of public, but you would also be able to see what's shared with you via the shared with me that link, and you'd see the collection of files or folders. Give that a second to load. As I said, my wireless unfortunately is a little bit slow today, but it doesn't really impact anything too badly. fact, what I'm going to do is just go and show you one or two more quick features and have to move on to actually analyzing data. So let me come back to my home folder, and I just wanted to share up and upload. I just wanted to point out that you also have the ability to import files from a URL. So this would be as simple as pasting in a URL that you wanted to share with somebody. Also here you have the ability to download files or folders. So if you were, I'm just going to go ahead and close that and reload that. If you were, if you had some files or folders, and again, I don't recommend this for things that are very, very large, but if you had something that you wanted to download, you could do that there. Via metadata, the other thing that I wanted to emphasize, I really don't have time to do it justice, but everything within the system here is actually labeled with metadata. That gives you the ability, for example, let's say I have a file here, and I go to metadata and I want to edit or view the metadata for that file. This file probably doesn't have anything all that particular attached to it, but I could arbitrarily add metadata. So I could say an attribute is something like, let's say time point. And I could say this is the time point 1, and this is unit is hour, so 1 hour into the experiment. This would be an attribute that I could add, and I could say this one is, experimenter, and this is Jason, and there is no unit attached to that. When we have files, or even if you could see on this particular file that it has a name like Arabidopsis_Thaliana, TER 10.26, cDNA, ultimately we can't name our files intelligently forever by just adding underscore this, dash that, version 1, version 2, version 3. But by attaching metadata to it, we can actually have those information associated with the file. If you look back on the screen here, obviously what I'm doing is quite tedious to do by hand, and you couldn't do it in bulk. So one, we have a template. For any individual template, let's say we were going to submit something directly from the discovery environment to NCBI, an NCBI biosample, if we take a look at that template or download that template, let me just go ahead and extract that. There is a sort of a guide as to how those data would be filled out. And then ultimately, you may have a CSV file that contains all of the information that's required, and you may have a collection of data, and you can actually pair those things. You can upload your template and upload your files, and you can pair them together so that you actually have a collection of data that's really well described according to best data practices. Okay, so I'm not going to save those changes. When you have things labeled with metadata, it actually becomes easier to search. I don't have to search by name, which is, could give me what I want, or maybe it won't. I could actually go up to this smart search where I click this little funnel and say I want to search for things that have the attribute genus and has the value Dioscorea. Maybe I want it from a certain size, or I want it modified or owned by somebody, or et cetera, et cetera, et cetera. But you have a much better way to search through data sets because they are labeled according to a really flexible metadata scheme. Okay, so I'm not going to go through metadata and searching just now, but I wanted to point out that that exists. Okay, so we have to move on because we actually have to talk about how to analyze data. Just going to give you a quick idea of what my goal is here in order to do this demo. We're just going to do a really simple analysis because if you understand how this works, then you basically understand how to analyze any type of data set within the discovery environment, or the DE as we call it. We're just going to take some unaligned DNA sequences, which are in a FASTA format, and we're going to create a multiple sequence alignment. So let's get started. That's just going to be a quick analysis that gives you an idea of the capabilities here. So if I click on apps or applications, that opens up, okay, a whole listing of the different applications that are within the discovery environment. As you can see, it's actually nicely organized. So if I want to go and take a look, I have some different types of categorizations of my applications. If I'm looking at something, like genetics, or maybe I want to go ahead and do a GWAS, there's different ways that these things are organized. If I wanted to say, look at alignment, oops, I wanted to click the first one. you at least have some ideas of how the applications are analyzed. And so ultimately, I'm just going to go ahead and search for an app. I'm going to go ahead and search for muscle. And I want to use this muscle 3.8.31. If you look, there's sometimes multiple versions of apps. You may not know what an app does, so you can click a little I info button. It gives you an idea of what that application does and who created it. Usually we just wrap those applications here. We haven't authored those. Sometimes there's a rating assigned by the community. You can leave comments if things are working or not. But to use an application that you search for, I'm just going to click on the name of the application and it's going to open up this window. Okay? And from this window, I have certain things I need to do. I need to give my analysis a name, so I'm just going to call this underscore September 2016 underscore demo. And I'm going to leave some comments. This is a demo for 2016 webinar. I can choose where that output result will go, but usually by default it's my analysis folder. I need to select some data, so I'm going to go ahead and browse my data store. It automatically opens up to the last place where I was looking for something. In this case, let's see if it gets it right, because my data are in home training DE walkthrough, so it actually looks like it's going to the sort of right place. And in there is a file that I'm going to look at, DE sample plants. And this is just some unaligned sequence data, so I'm going to go ahead and say OK to that. It's going to ask me what my sequence type is, and in this case I happen to know it's DNA. So all of the applications have this sort of step-by-step where you move through what's needed to use that application. I'm going to go ahead and click Launch Analysis. Once I click that, I'll get a notification up top that my analysis has been launched, and then I just have to wait a few moments in order for it to complete. So let me just let that complete there. Get some of this out of the way. And when that does complete, I'm going to go ahead already and open up Analyses. Okay, there's my notice that that's been launched. I clicked on this other button. If you didn't see it, let me do it again. Analyses. So this is Analyses. I'm also getting feedback on the right-hand side that demos are running and things are happening, so that's good. And this is, Analyses is important because it is a history of the applications that I've run. So if I want to see things using the Muscle app that I've run before, or things using Top Hat that I've run before, I can see a history of those analyses, okay? If I want to look, some analyses might be shared with me, so I can actually share things. If I've done an analysis and I want, hey, you know, take a look at these results, I can click on Top Hat, and I can go ahead and share this with another user, so that's really important. If I go up to Analyses, there's a variety of things that I could do there too. So I have a job that's running. Maybe I decide, wait a second, I shouldn't run that job. Maybe I need to cancel that job, so I can go to Analyses and cancel it if it hasn't completed yet. I can also go ahead and view some information about that analysis. Right now it hasn't completed, so there's really not much there. But if I go ahead and take a look, let's say I go to a Top Hat job that I did and I go to Analysis and say View Analysis Info. Whoops, not Analysis Info, but actually Analysis Parameters. I can actually see all of the settings and everything that I did previously. So it's really like a lab notebook because I can see what files I used, and I could actually click on any one of those files and be taken to the raw file. I can see all of the settings that I use. So this is tremendously important. If I wanted to, I could even export that by saving that as a simple text file wherever I'd like to save that. Okay. As you saw, doing this point and click, if I have a job like, or an app like Top Hat, that could be a lot of pointing and clicking in order to rerun that analysis. So I can go up to Analysis and actually just say relaunch. And then what that will do is it'll actually launch the Top Hat app, except this time, everything that I had used before, all of those settings are the same as what I had used the last time. It's going to be pre-populated with all of those settings that I had used before. So I could go back and I could just say, well, instead of 8, I just need to change this one setting and make the anchor length 7. So that's a big time saver. Okay, so let me go back to Analyses. Okay, right now this is still running. It might take a few more seconds. If you're not sure, you can always click refresh, but I didn't see the notification that it's done. It will finish up in a second, but when your job is done, you can actually click on the name, and it looks like I did one before. So you can click on the name of that job, and that will actually take you directly to the output of that job, so you don't have to go searching where did the results go. So if I give a second to load, it will take me and I'll be able to actually see the results of any previously completed job, and I can download those, I can share those, I can do whatever it is that I need to do in order to interact with that job. Okay, so just give it a second to load and we'll see the results. Back here, you also, some things I didn't cover, you have the ability to rename those job names and update some comments. So if something was particularly useful or not, you can note that there. You could also have the option of deleting any history that you don't need. Okay, so that is making its way towards that. Again, my reception is not the best here today. Otherwise, it would be a little bit faster. So I'll give that a chance to load, and in the meantime, I'm just going to keep moving. So we get through, we only have a few more minutes. Okay, I just got a notification actually that my other job has completed. I can open that one up too. Let me just refresh since it's done. Okay. And I think this will also open up in a new window. Okay, so when it's done, it will load, and in this case, I have the files that are the result. If I were to click on that, and this is just because I'm out of sync here with this. Let's try this one because I'm out of sync with my Wi-Fi here. Those are the results, and you'd be able to actually share them, preview them, edit them in any way that you like to. But in this case, it's really just these aligned sequences. So I'm going to go ahead and move on to the next demo that I have to cover. And that demo is actually with cloud computing. In fact, it's the last demo because we've covered data and we've covered analysis. So Atmosphere, as I might have mentioned before, is actually a cloud computing setup. And I'll remind you also, I don't see any questions yet, but if you do have questions, please feel free to at any time let me know about them. Atmosphere is a cloud computing platform. Many biologists, bench biologists, may not know much about cloud computing in the life sciences, or they may be experts, but let me cover the basics with a couple of analogies. First, I need to start out with a term that you need to know to understand this. One is something called an image, and the other term I'm going to talk about is something called an instance, just to define what is cloud computing. So let's just take the circumstance. Let's say you bought a new computer and you need to move files from your old computer onto your new computer. You're probably going to take those files and put them onto a hard drive. So you copy them over, now you have a backup. But there's another type of file that you could make from your original computer, and that's called an image. And an image is a copy of everything on that computer, not just individual files or folders, but actually the operating system, the settings. It's the most complete representation of that operating system. So if you take just ordinary documents and pull them off a hard drive, you still have those original documents. But if you take an image out of a hard drive, you can actually completely clone both the files and the data, the operating system, the settings, even the desktop background onto a new computer. So that's what an image allows you to do. It's really everything you need to run a computer. The next concept is something called an instance. So in the CyVerse cloud, we have hard drives, CPUs, memory, RAM, right? And when you come to CyVerse, you'll say, well, I need a certain amount of this in order to do an analysis. Let's say you need several CPUs to do a fairly sophisticated assembly. Well, you'll come to us and tell us how much you need. You're also going to need software to run that, so you'll need an image. And when you put those two together, you have what's called an instance or virtual machine. This is a computer that you've literally just created temporarily, and you can access it at an IP address. When you come to Atmosphere to use it, you'll find that we have up to 16 core, 128 gig instances available, and you can access those via shell or VNC. You can use an instance that we have existing, or you can make your own. You can share your modified instances with the public or just with your lab members. Some of the use cases for using Atmosphere include things like running the software and data sets, the analyses that you right now have to dedicate a single laptop to. You can't use your laptop for anything else when you're running some other type of analysis. You can run programs that have a graphical interface. You wouldn't be able to do that in the discovery environment. With Atmosphere, you have full administrative, or what we call pseudo access. You also can use Atmosphere to generate computing setups for your lab. So, pardon me, you could have a set of software and settings and parameters that are enshrined in an Atmosphere instance, and then you always have access to that machine so that anyone using the same instance is using exactly the same setup with all the parameters, all of the dependencies that you've originally configured. People also use Atmosphere to distribute their own software. So let me give you a quick demo. Our demo is only going to be launching an instance and then connecting to it. As I mentioned, if your instance, if you want to connect to it, you can use either PuTTY in Windows or any type of shell or terminal access you have in Mac and Linux. If your instance has a desktop, you can also use VNC Viewer to use the desktop feature. And I'm going to do both of those. So let me take a look. Let me just close this discovery environment stuff for the moment. And back on the home page of CyVerse, you could click on Atmosphere and log in. It would bring you to this page. So this is Atmosphere. In fact, let me log in because I'm just viewing instances. When you log into Atmosphere, maybe the first thing you'll want to do is launch a new instance. Here, since I'm an active user, I have some history. I see some idea of the resources I'm using, but I can go ahead and click on Launch New Instance. Then I might search for topic. Let's say I'm looking to do RNA-seq. I could put in that topic, and then I could see various instances that are tagged with RNA-seq and get some information on that. I could click on an instance that I want to run, and then I click on Launch. I choose the size that I need. Now, of course, there's some limitations here. Sometimes the system may not have all of these available, all the sizes available. And you also have a sort of limit to your resources. So if I go above that limit, I won't be able to launch. In fact, I'm not quite above it yet, but I do have the ability to ask for more resources. So you'll see those links everywhere. But once you've selected your setup, it's a matter of clicking Launch. Now, I already have things in a project that have an active instance. So if I click on this, or if I look, once I launch it, it takes about 10 to 15 minutes for it to become active. But once that light is green, I can connect to it. I've got an IP address. If I click on that actual instance, I've got some more details. I'm going to go ahead and copy that IP address. And since this has a desktop, I'm going to go ahead and open up this VNC Viewer, which allows me to connect to things that have a desktop. Type that in and hit connect. I'll say yes, it's okay to connect. I'll enter my CyVerse username and password. And this is my instance, and I must have it set to a low resolution here. Let me adjust that, see if I can make it look better. Okay, there we go. Looks a little bit better than the 8-bit color. So I'm connected to an Atmosphere instance, and this is essentially the desktop of a computer that's at my disposal. I haven't used it for anything else, but I could use the software that's installed there. I could install my own software, and it's basically a computer that I just got off the shelf and operate. If I wanted to connect to it via the terminal, I could start a terminal here. I could use SSH and then do my username at that IP address. In this case, I've connected to this one before, so you'll never have to do this, I believe. Let's just open this. I will just edit this one, since it's just a little bit of a security thing I have going on here. And let's just get rid of these. Okay. Let's try connecting again. There we go. Say yes. Enter my CyVerse credentials. And now I am going to be connected. Oops. There we go, to Atmosphere, also the same computer. If I wanted to, I could make a directory. And there's going to be a new directory on that computer. So this is essentially a computer. I can use it to do anything that I would normally do on my laptop or another server. But the point is that I may have data sets installed on this image. This computer may have a lot more RAM, a lot more CPUs, a lot more memory than my desktop or my server, and it's on-demand computing that I can do to analyze. I can share this with another user. They could log in with me. We could work through an analysis together. So it's really flexible and quite powerful for folks to use. They even use it in the classroom where you might have several users or a bank of Atmosphere instances running where your students connect to it. So it's quite powerful. Let me go back here to show you what you need to do once you're done using it, because these are finite resources. So once you've finished using your Atmosphere instance, you could either stop that instance and just pause it to come back to it another time. You could also, if you've made changes and you want to make those changes permanent in a new image, you could re-image it by clicking that. But what I'm going to do is I'm going to delete it because I no longer need it. Now let me make one important caveat. When you start an Atmosphere instance, it is not connected to your data store. So it doesn't, and it wouldn't make sense to copy all of your data into this automatically. You have to connect it and tell us what data you want to bring in here to analyze. But once you're ready to end this, you need to transfer that data back to your permanent data store or move it somewhere else. So once you are ready and you've saved your data, you can come back and delete. It'll warn you that any data that's on there that's not saved elsewhere to a permanent machine will be lost. So I'm comfortable with that at the moment. I'll go ahead and delete it, and then that will start to spin down. Okay. As I mentioned, and I'll click back here in your dashboard, you have a finite set of resources that you could use at any given time. If you need more, you can simply click on that link and file a request. But it's really pretty flexible and pretty powerful. Okay, so we just have 2 more minutes here. And whoops, I did not want to do that. Not my email. I wanted to come back to the PowerPoint and just close this down here. So where to go from here? We recommend that you go back and check out the Learning Center on the CyVerse website. You'll find tutorials and documentations. You'll also see under upcoming events additional webinars and workshops. You can also ask us for some training, whether you would like a workshop and we can come to you, or you'd need a webinar or some other support. We're happy to do that. We also want to acknowledge, at least the executive team, there's many more people that make this up, but across the various sites who help make CyVerse what it is.